Hello, future nurses and science students more, and all those doing health studies. We are bacteria, and we are the microscopic, single celled organisms that keep you on your toes during every single clinical rotation. If you look at us under a microscope, you won't find a nucleus, a nuclear membrane, or complex organelles like mitochondria inside our cell walls. In the biological world, that makes us prokaryotes. We are incredibly simple, yet we are masters of survival, adaptation, and reproduction. In your nursing courses, especially when studying infection prevention, you will spend a massive amount of time learning how to kill us, stop us from spreading, and treat the infections we cause. But here is a fun twist: we aren't all villains. While some of us are dangerous pathogens that cause severe disease, millions of us live peacefully inside your body right now as your beneficial normal flora, like the bacteria in your gut that help you synthesize vitamin K. Let's break down our structural blueprint so you can ace your next microbiology or pharmacology exam. Shaping up the three major bacterial morphologies. When a laboratory technician takes a sample from your patient's wound or blood culture and looks at us under a microscope, the very first thing they note is our physical shape morphology. We fall into three classic categories. One, Kochi, the spheres. We are round, ball-shaped cells. How we bunch together tells a huge story. Staphylococci, we bunch together in irregular clusters, looking exactly like a bunch of grapes. Clinical example, Staphylococcus aureus, the culprit behind skin infections and MRSA. Streptococci, we line up perfectly in long straight chains. Clinical example, Streptococcus pyogenes, the cause of strep throat and rheumatic fever. 2. Bacilli, the rods. We are elongated, brick-shaped or rod-shaped cells. Clinical examples: Escherichia coli, E. coli, which is the number one cause of urinary tract infections, UTIs, and Mycobacterium tuberculosis, which attacks the lungs. 3. Spirilla, spirochetes, the spirals. We are wavy, corkscrew-shaped cells that move with a twisting motion. Clinical example: Treponema pallidum, the bacterium that causes syphilis. Comparing the three main shapes of bacteria, Kochi in clusters, chains, bacilli rods, and spirochetes. The ultimate shield, gram-positive versus gram-negative. This is the most critical concept for your nursing pharmacology classes. To choose the right antibiotic, doctors must know the structure of our cell wall. We use a diagnostic coloring test called a gram stain to divide us into two massive camps. 1. Gram-positive bacteria, the purple team. The structure: we possess an incredibly thick, heavy outer layer made of a sugary protein mesh called peptidoglycan. The stain: when the lab tech applies a purple crystal violet dye, our thick peptidoglycan wall absorbs it like a sponge and holds onto it tightly. Under the microscope, we stain a deep purple blue. Pharmacology connection. Because we have this exposed peptidoglycan wall, broad-spectrum penicillins and cephalosporins can easily target and shred our structural barrier, causing us to lyse and die. 2. Gram-negative bacteria, the red-pink team. The structure: we are much sneakier. We only have a very thin layer of peptidoglycan, but we protect it by wrapping it in a tough, extra outer membrane made of lipopolysaccharides, LPS. The stain: when the purple dye is washed away, our outer membrane blocks it, and we absorb a counter-stain dye instead. Under the microscope, we stain a bright red or pink. The endotoxin danger. This is a massive nursing alert. That outer lipid membrane is also known as an endotoxin. When an antibiotic kills us and rips our cell wall apart, we release that endotoxin into the patient's bloodstream. This can trigger a massive inflammatory response, causing blood vessels to dilate violently and leading to life-threatening septic shock. Image comparing the thick peptidoglycan wall of gram-positive bacteria with the double membrane lipopolysaccharide wall of gram-negative bacteria. The secret weapons, capsules, and spores. To survive your patient's immune system and the harsh environment of a hospital room, some of us can develop specialized armor. 1. The capsule, the invisibility cloak. Some bacteria can secrete a sticky, slimy sugar coating around their outside wall called a capsule. This capsule acts like an invisibility cloak. When frontline responders like Ned the neutrophil or Max the macrophage try to grab onto us, the capsule makes us too slippery to swallow, allowing us to multiply unchecked. 2. Endospores, the ultimate survival bunker. When conditions get tough, like when a nurse sprays alcohol on a surface, or when there is no water or food, certain rod-shaped bacteria, like Clostridium difficile or Bacillus anthracus, can pack their DNA into a hard, indestructible, dehydrated shell called an endospore and enter a state of suspended animation. The challenge for nurses: endospores are completely resistant to heat, drying, alcohol rubs, and standard hand sanitizers. Infection control action. This is exactly why, when caring for a patient with C. diff, you cannot just use hand sanitizer. You must physically wash your hands with soap and warm water to friction rub the stubborn spores off your skin, and you must clean room surfaces with bleach to melt the spore shell. Critical nursing implications and infection control. As a nursing student, managing bacterial infections requires applying your knowledge of microbiology straight to the bedside. Empiric versus definitive therapy. When a patient arrives with signs of sepsis, a nurse must draw blood cultures immediately before hanging the first bag of antibiotics. The lab will perform a gram stain to give an initial clue. Empiric therapy. Once the culture grows and identifies the exact bacterial species and what drugs kill it, sensitivity testing. The provider will switch to a narrow, targeted antibiotic. Definitive therapy. Antibiotic resistance. If a patient stops taking their antibiotics early because they feel better, they leave behind the strongest, most mutated bacteria. These survivors share their resistance genes through tiny tubes called pili, conjugation, creating deadly superbugs like MRSA, methicillin-resistant Staphylococcus aureus, and VRE, vancomycin-resistant Enterococcus. Always educate your patients to finish their entire course of antibiotics. Follow, like, and share for more.